Okay, here's something done by a proper artist rather than from my hokey cutaway drawing from the last slide. Um, you can see the model of the sponge, which has pores. And remember, these are the internal bit of a donut or tube-shaped cell called a porocyte. Okay, so you can imagine this is a cutaway right here. So you have to imagine that that is like a tube, and it would go all the way around. And right in here is where the water can come in through from the outside to the inside of the sponge body. That's called the porocyte. And then, of course, the water will go out through the osculum. Oscula, plural, are the holes you can see in the sponge. Okay, so you cannot see where the water comes into a sponge. Remember that it's only one cell. And then we see the choanoderm, where the choanocytes are lined up on the inside of the body of the sponge. And with this drawing here of a collar cell or a choanocyte, the reason they're called a collar cell is because they have this collar of cilia. So the flagellum here beats, and it induces a water current, and that water current also brings water through this collar of cilia, and then the food particles get stuck on this that's sticky, and what happens then is the amoebocytes and the collar cells will both ingest these particles into vacuoles within the cell and digest them. So we also see the pinacocytes and the pinacoderm. So the pinacocytes are lined up on the outside of the outside lining of the sponge, like a protective layer. We have amoebocytes or wandering cells here, which wander around and share food and also secrete the spicules. And here are some shapes of spicules. You can see that they have different shapes depending on what species they are, and we can actually use those to identify which species of sponge that we're dealing with sometimes. So I'm not going to linger on these slides, but you can stop and use these as notes to cover what we've already told you, but these are a more refined definition. Same with this slide. Okay, sponge self-defense, we covered it a little bit in the last video, but remember we said that a sponge sitting on the bottom or any living piece of any living material that's attached to the bottom can't run away, can't go anywhere. It's essentially a bag of meat. It's just meat sitting there. And what happens when you throw something like that into the ocean? Something will come along and eat it. So how do they defend themselves? They have a lot of toxins, and this is why we can't eat sponges in general, because they are full of poison compounds in order to stop things from grazing on them. They also have warning coloration, so lots of sponges are brightly colored, and you think, why do you stand out? Why do you want to stand out if you're a bag of meat when you want to hide and not be very visible? Well, if we think about something like an insect with yellow and black stripes, are you going to go up and try to catch it or try to eat it or anything like that? You're not, because you know that based on that coloration, you're probably going to wind up with something painful happening. So warning colorations are meant to scare predators and grazers off. And then finally, the spicules. Spicules, these calcium carbonate or silica fibers, shapes within the sponge. You can see here they're kind of like the palisades on a paw. They're defensive, they stick out, and they're also structural as well. Obviously, these things are like the girders or the framework of a house. But when you think about being a predator or a grazer, if you come up and take a mouthful of this kind of thing, then what happens? It's like chewing on needles, and that's not going to be very pleasant. Here's a micrograph of some types of spicules. There's another micrograph of them, and we can use both the arrangement, how they sit within the body of the sponge, and the different types of spicules. And all of these spicules all are different types of spicules, different shapes that you'll find within the same type of sponge. So this one is a black pillow sponge, but there are probably 7 or 8 species that look very much like this black coloration and similar body shape. So it's very difficult to just glance at a sponge and see what species it is because they don't all grow into the same shape. Each member of a species doesn't grow into the same shape as other individuals of that species. So sometimes spicules are the only way to diagnose these. This is the spicules and spongin as well. So spongin is a protein compound that is more like a it's the squishy type of sponge. It's a bath sponge. It's very flexible, but it's very structural. So it doesn't provide us like a defense mechanism. It's essentially just a building block. And orange golf ball sponge and the arranged golf ball sponge. Uh, Anchorina alata. This is another local sponge. It's yellow generally. This is another type of. These are all. That's breadcrumb sponge, pitcher sponge, and those are the arrangement of and type of spicules within that particular species. These are all ones that you'll be familiar with after diving around New Zealand. Elkhorn sponge and another low nipple. Another. Okay, so what are spicules made of? They're made of either silicon dioxide, which is glass, or calcium carbonate, which is the type of material that will be in a seashell or bone, very common, biogenic, made by living organisms. Biogenic means made by living organisms. Biogenic, generated from biology, another way to think of it. Okay, and you have the protein spongin fibers, as we were talking about before. So some of these, because they are essentially silicon dioxide, conduct light just like fiber optic cables. Okay, so here, if you can do something where you dissolve away all the organic material, and this is kind of what spongin looks like. And we'll get a chance to look at that well, hopefully we'll get a chance to look at that in the lab. But this is what you're left with if you have a sponge and you dissolve away all the living material and you wind up with just the spongin. So this is kind of like the cosmetic bath sponge type material. All right, there are 3 structural forms. Asconoid is the simplest. Ciconoid is a sort of a medium one with a little bit of thickness to the body, and the leuconoid are the common ones that we generally know as sponges, which are the bigger sponges. Remember, we were only talking about 2 cell layers thick in this diagram. So the choanoderm on the inside, the pinacoderm on the outside, and a little bit of a separating layer in the mesohyl with the amoebocytes in there between them. But most sponges that you see are quite big and, well, not quite big, but bigger than a cell layer thick. And so what we do is these structural forms are essentially a measure of how much folding of the tissue there is. Sorry, sponges don't have tissue of the body there is in order to produce a bigger sponge. So asconoid sponges would be ones that would be like encrusting sponges found a cell layer thick on a rock or something like that. And they're rare. the leuconoid sponges are the most common. But when you say that you can add surface layer with the ciconoid by doing this folding, and then it gives you much more space for um, choanocytes, and the number of choanocytes are essentially the amount of feeding that you can do. So if you have a lot more choanocytes, so as the volume goes up, so if you know that, if you know that the volume of a sphere goes up much faster than the surface area of a sphere, then that's what happens. In the inside of the leuconoid sponge is packed with choanocytes, but they have less um, surface area in order to that make pinacocytes that can't feed, and so feeding becomes much more efficient as you go from the large or the smaller to the larger sponges. Okay, so again, this is I won't linger on these. You can read these, stop these and read the slides. There you go. And here is a 3D version of what a cutaway version of what a leuconoid sponge would look like, where you see these flagellated chambers or these chambers covered with choanocytes, and those choanocytes do a lot of filtering. And so by having a bigger body with lots and lots and lots of these chambers, then you can be much more efficient. So this is the last of the slides for this video. We'll see you in video 3 of Porifera. I would go to the cycnoid and leuconoid sponges on page 22 of your theory book and review. I would also say that there are many good animations and other explanations of how sponges work, which I've provided links, which should help you review on Moodle. And if you type in things like "how does a sponge work" into YouTube, you'll get some pretty amazing videos. Some good David Attenborough and the like. Okay, bye-bye.